Hello everyone. Today we are seeing what is antigen and what is antibody. First, what is antigen? Antigen and antibody are the key components of an immune system. They play an important role in a body defense against the pathogen, which includes the virus, bacteria and other pathogens. An antigen is a molecule or a substance that can be recognized by the immune system and as a foreign or it is a non-self. The antigens are typically act as a protein or it is a large polysaccharide that is found on the surface of the pathogens such as the viruses, bacteria and parasites. They can be also present on the surface of the cell from the transplanted tissue or even on a cancerous cell. When our immune system is detected an antigen, it triggers the immune response to eliminate or neutralize the threat. The antigen are again classified into two types: foreign antigen and self antigens. That first, it is a foreign antigens. What is foreign antigens? The antigens which are derived from the outside our body. And in other hand, what is self-antigen? The antigen which are derived from the individual ones, or they are recognized as a self by immune system. The immune system usually does not mount an immune response against the self-antigen unless they are altered or presented appropriately, which can be leads for the autoimmune diseases. Next part, it is antibodies. What is antibodies? The antibodies is also known as the immunoglobulins. Immunoglobulins, it is a proteins that are produced by the specialized white blood cells and B lymphocytes. The antibodies are specially designed to bind to the antigens and neutralize to mark them or destroy by each other components of an immune system. Each antibody is highly specific to a particular antigen, and the body it can be produces vast array different antibodies to recognize and combine the different pathogens. Then we see the structure of an antibodies. How the antibody will look. Antibodies have a Y-shaped structure which is composed of the two heavy chains and two light chains. The tip of Y-shaped structure contains the variable regions that can be bind to the specific antigen. It is a binding site of an antigen. This binding occurs through the non-covalent interactions such as a hydrogen bonding, electrostatic attractions. Once the antibodies binds to the antigen, it can be initiate the several responses. How the antibody will be produced? The production of an antibody is a dynamic process that involves the activation and differentiation of an B cells into the plasma cell, which is the antibody secreting cells. This process is initiated between the B cells encountered an antigen that matches their specific receptors. Once the activated B cells are undergoes the clonal expansion, producing the large number of plasma cells that secretes the antibodies. Some beta cells are also differentiated into the memory beta cells, which can be provide the long-term immunity by the quick responding to the subsequent encounters with some antigen. In last, the antigen it is a foreign or a self substance that trigger the immune response, while antibodies are the proteins which can be produces the B cells that are specifically bind to the antigens, and they plays the important role in neutralizing and elimination of pathogens. The interaction between the antigen and antibody it is a fundamental aspects of an immune system, and it plays a crucial to protecting the body against the infections. Here we see what is the antigen and antibody. Thank you.